Thank you, everyone, for attending. As you can see, the title of my lecture is Misfolded Proteins in Neurodegenerative Disease, Molecular Consequences and Cellular Decompensation. And what I'm going to try to do in this lecture is, first I'm going to provide a bit of an overview of this field in terms of the inherited neurological diseases that we study in my lab and how this fits into the broader field of neurodegenerative disease, an important area, especially as the population ages. Then after that introduction, I'm going to tell you two vignettes about research from our lab. We have many projects going on, so I want to apologize, especially because I know there are people from my lab here, and I won't get to talk about their work. We have a large research program, so I will limit my lecture to just these two vignettes, and one will be about a disorder known as Huntington's disease, and the other vignette will be about a disorder known as Spinocerebellar ataxia type 7. So, okay, well, without further ado, we will begin. So, in terms of thinking about the history of the neurodegenerative disease field, people talk about the first golden age in the study of neurodegenerative disease as occurring about a decade ago. And indeed, provisional clinical descriptions for all of the major neurodegenerative disorders were in place well before even the 19th century. And then what happened was that pathologists began to study central nervous system and brain material at autopsy. And basically, due to advances in microscopy and the application of new staining techniques, there was fervent activity in this area. And what happened was these diseases became classified as distinct clinico-pathological entities. And physicians such as Dr. Alzheimer, Dr. Pick, Dr. Louis, and others were intensively involved in this effort. And this important work took place at the beginning of the last century, roughly. And of course, a number of the disorders or the abnormal findings bear the names of these individuals. So after that important set of work that was done more than a century ago, really, I think the next important era of discovery took place about 20 to 30 years ago, and this was fueled by advances in molecular genetics. And basically, this work, which really involved the tools of molecular genetics that we still use today, involved a process that relied heavily on clinicians, because this required the collection of families, patients in families that were suffering from a variety of inherited neurological diseases. And there are individuals here at this institution, such as Dr. Tom Bird, who were involved in this effort. And once the families were collected, what was done was that disease genes were localized using an approach known as restriction fragment length polymorphism mapping. And in this way, genes were localized to particular chromosomal regions. And then within those chromosomal regions, candidate genes were sought. And those candidate genes were analyzed until mutations were identified. And in this way, the genes that cause different disorders were discovered. And a lot of this work took place in the 1980s and 1990s and has led to a lot of information in terms of the genetic basis of inherited neurological disorders. And this has allowed us to subsequently work on developing animal models, studying disease pathogenesis, and there's also clinical implications for this work in that now we can do genetic counseling and offer diagnostic tests. And indeed, Dr. Tate, who runs our molecular diagnosis unit, has been involved in the development of the testing modalities that we use in the clinic as it applies to this disease field. Now, what's left to do is to figure out what's going on in these diseases, and there's still a lot more work to do. But something that we have made very little progress in, unfortunately, is in the area of treatment. And that's really hopefully will be the next era of discovery that we embark upon soon. Okay, so like all good stories or genetic stories, this one begins with a very rare disorder known as X-linked spinal and bulbar muscular atrophy. It's also known as Kennedy's disease in honor of the neurologist who described it back in 1968. And this is a disorder that is characterized by a lower motor neuron disease. It's a disorder that only affects males. And these patients get proximal muscle weakness, they get bulbar weakness. Bulbar refers to the muscles of speech and swallowing. They get atrophy and fasciculations. They do not get upper motor neuron findings, so the disorder can be reliably distinguished on clinical grounds from amyotrophic lateral sclerosis. It's a progressive degenerative disorder, and what is particularly interesting about this disorder is that patients have signs of androgen insensitivity, including gynecomastia, as you can appreciate here in this patient. Now, the signs of androgen insensitivity were an important clue in terms of what might be the cause of this disease, and it led us to consider the human androgen receptor gene as a candidate gene for this disorder. And what I'm showing in this slide is that the human androgen receptor gene contains a trinucleotide repeat track, so 3 nucleotides, in this case CAG, that are repeated over and over. And this actually falls in the protein coding region, so CAG would encode the amino acid glutamine in the androgen receptor protein. And we were drawn to the androgen receptor as a candidate for the clinical features that I explained, and also because of the X-linked pattern of inheritance. And so when I was a graduate student in the laboratory of Kurt Fischbeck, we looked at this gene in patients with SBMA, and what we discovered is that patients with this disease have an expansion of the CAG repeat track. And the way this works in this disorder, and this theme applies to most of the repeat disorders that we'll discuss today, as well as most of the repeat disorders in this entire field, is you have repeats that are variable in their length in normals. However, in patients who have the disease, you have an expansion. So you have a separate, non-overlapping distribution of expanded repeats. And so what you see in this disease and in other diseases of this type are that this is an absolute association between repeat expansion and disease phenotype. So everyone who has the disease has a repeat expansion. Everyone who doesn't have the disease has a repeat in the normal range. And for this reason, we can offer molecular testing that is highly specific. Okay. So since that discovery in 1991, it's become clear that repeat expansions are a common cause of neurological diseases. And this slide summarizes the list of disorders that are caused by repeat expansions. And what I'd like to point out is some of these disorders are rather important because they're common. Fragile X syndrome of mental retardation is the most common inherited form of mental retardation. Myotonic dystrophy type 1 is the most common form of muscular dystrophy, and Huntington's disease, as we'll discuss, is one of the most common inherited diseases known to man. Now, these disease repeats, I just want to discuss very briefly a little bit about them. They're very interesting. Most of them are trinucleotides, as I showed you for SBMA. However, you also can have tetranucleotides, pentanucleotides, or even dodecamer repeats that are expanded to cause disease. And repeats of this length are known as microsatellites. Now, it's interesting because these types of repeats are very common in our genome, and people have known about them for half a century. And initially, they were thought to be of no functional significance. They were studied for years as an evolutionary curiosity. But when, in 1991, repeat expansions were discovered as a cause of human genetic disease, this created a third major class of molecular genetic mutation. And so repeat expansions stand sort of in between, the gross rearrangements that you can see at the cytogenetic level and the very minor alterations that you would detect by DNA sequencing. And in terms of classification of these different repeat diseases, what has been useful is to look at the sequence composition of the repeat and also the location of the repeat within the gene. And using this approach, we've been able to classify 4 different types of diseases. And as you can see, the repeats can be located in a variety of different places, in the coding region, in the promoter region, in introns, or in the 3' untranslated regions. And so the 4 types of repeat diseases that are now recognized are listed here. The first class is loss of function repeat diseases. These are repeat diseases where expansion of the repeat prevents the expression of the gene within which the repeat resides. And the Fragile X syndrome of mental retardation would be an example of such a disorder. The CAG polyglutamine repeat diseases are disorders where you have an expanded polyglutamine tract that's made in the protein, and that causes the disease. Then there are RNA gain of function repeat diseases where the problem is that you make an RNA that has too many CUGs in it, and that leads to dysregulated splicing, and so you get a disease. And then finally, there are a group of disorders where the repeat encodes the amino acid alanine, and you get too many alanines, and that's the problem in those disorders. So there are these 4 classes of disorders, and the research in my lab has focused upon the CAG polyglutamine repeat diseases. And this class of diseases, repeat diseases, consists of 9 inherited neurological disorders. We've already discussed spinal and bulbar muscular atrophy. This category also includes Huntington's disease, which, as I said, is one of the most common of all inherited diseases known to man. There's a very rare disorder in this category, dentatorubral pallidoluysian atrophy. And then there are 6 forms of spinocerebellar ataxia. And these diseases form a category because they share a number of features. They all affect the nervous system. They all are caused by dominant mutations. They all show dominant inheritance except for SBMA, which is, as I said, X-linked in its inheritance pattern. Median ages of onset are comparable in the different diseases. They're slowly progressive disorders. And then they all share this very interesting genetic feature where there is a correlation between increasing expansion size and disease severity. So as the repeat expansion is larger, the disease gets worse. And it's this correlation plus the tendency of the repeats to expand when they're transmitted from parent to offspring that explains the clinical phenomenon of anticipation, which was described in 1918 by a German physician studying patients with myotonic dystrophy. And so what was observed is that as you went from one generation to the next, the disease became much worse. And then the final shared feature, the critical feature, is that they're all caused by CAG repeat expansions. But these CAG repeat expansions always fall in frame to encode glutamine. You wouldn't expect that by chance. They could fall in frame to be AGC or GCA, but for these disorders, it's always CAG, so it's always a glutamine expansion. Though there is one notable exception. SEA6 is admittedly an oddball member of this category of diseases because you can get this disease by having point mutations. Okay. So then what is the molecular pathology in these disorders? Well, as we discussed, you have too many repeats at the DNA level, and what happens is then you make a protein that has too many glutamines. Well, why is that bad? Well, the reason why that's bad is then the protein misfolds. And we know that it is the misfolding of the protein that is the crux of the pathology in these disorders. So there is a discrete threshold that cuts across these diseases at roughly the same repeat length. And once you cross that threshold, then you make a protein that adopts an abnormal conformation. And that is the key initiating event in producing disease in these disorders. Now, it became clear about a little over a decade ago that this protein misfolding phenomenon was not only important for these rare neurological diseases, but actually cuts across all of the major neurodegenerative diseases. And this recognition came from, again, ties us back to that work that was initially done a century ago by the neuropathologists who identified protein aggregates and named them, often after themselves, in these different diseases. So as we discussed in the polyglutamine diseases, there's misfolded proteins, and the hallmark pathology are nuclear inclusions, as I'll show you. In Alzheimer's disease, there are misfolded proteins that involve neurofibrillary tangles produced by tau or beta amyloid plaques. In Parkinson's disease, you have Lewy bodies, and that involves the protein alpha-synuclein. And even in ALS, there are misfolded proteins in the cytosol in the inherited version in particular that involves superoxide dismutase 1. And I think most of you will be familiar with the prion diseases because you've heard of mad cow disease, since our state bears the ignominy of being the only state in this nation to have a mad cow identified, though I understand the cow did come from Alberta, so it's really not our fault. We can blame the Canadians. But the main point I want to make here is that this notion of protein misfolding being a key concept in neurodegenerative disease, it was driven in part by work done on these relatively rare diseases. And now, as I lectured to you in 2008, what we strongly believe is that protein misfolding is a common link that ties together all of the major neurodegenerative diseases. And why neurons are exquisitely sensitive to protein misfolding is something that is very important for the research we're doing, and also in terms of thinking about the development of therapies. Okay, so now our research has focused on one of the key questions in the entire neurodegenerative disease field, again using these somewhat uncommon disorders as a stepping stone to figure this out. And that question is, what is the basis of selective vulnerability? So what do I mean by that? Well, shown here is a diagram of the central nervous system, and I've listed a number of different neurological disorders, all of which have at least some of them have inherited versions, some are exclusively inherited. And what you see is that they affect different regions of the nervous system. But for these different disorders, you have a mutant gene that produces a misfolded protein that's seen in all the neurons throughout the nervous system. And indeed, in ALS1, you have a mutant protein, superoxide dismutase 1, that's seen in every cell of the body. But yet in these different disorders, only certain types of neurons degenerate. And so the question is, why is it that the basal ganglia neurons degenerate in Huntington's disease? Why motor neurons in ALS? Why cerebellar and retinal neurons in spinocerebellar ataxia type 7? And so this is an important question, and a lot of our research is focused on understanding the molecular mechanism behind this selective vulnerability. Okay, so we're going to transition now to the research that we're doing. We're in Seattle, so we have to have a Gary Larson cartoon, I guess. This says, "Dear Henry, where were you? We waited and waited, but finally decided that." So I don't want to leave anyone behind, but we have to move on. So hopefully what I've told you about so far makes a little bit of sense. And so the first story that I'm going to tell you about is our work on Huntington's disease. And this is an autosomal dominant disorder, as I mentioned, very prevalent in the human population, as many as 1 in 10,000, perhaps in some regions of the world, as many as 1 in 5,000 individuals are affected. It's a progressive movement disorder, and so patients get this uncontrolled dance-like movements. So sometimes the disorder is called Huntington's chorea because choreiform means dance-like. Patients suffer cognitive decline. There is psychiatric problems. Onset is usually in adulthood, but it can be earlier or later. And the disorder is relentlessly progressive and ultimately fatal. And patients can die from the disease as soon as 10 years after onset. Sometimes they can live with it 20 or 30 years. The region of the brain that's affected is in the basal ganglia, and the medium spiny neurons of the striatum are affected, and so it's this region right here that's affected, the movement control center of the brain primarily. Also you have cortical involvement. There are metabolic abnormalities. That's something I want to stress. It's not often appreciated, but there's muscle wasting, impaired glucose tolerance, and patients have a hard time gaining weight even before they present with their disease. And unfortunately, there's no cure, let alone any treatment for this horrible disorder. Now, people have been thinking about the basis of Huntington's disease for some time, and research has been going on for many decades. The gene was discovered in 1993, and that has led to a flurry of molecular research on the basis of this disorder. And it's been a little bit confusing because, as it turns out, if you study many different cellular pathways, you'll find that they're altered in this disease, and so you're left with wondering, what's really going on. But I think that one could say that there are two pathways that appear to be very important in Huntington's disease. One of them is mitochondrial dysfunction, and there's wonderful data that has been generated by Flint Beal, who's back in New York City at Cornell, and he showed in 1993 that if you treat mice with a mitochondrial toxin, 3-nitropropionic acid, they actually get degeneration of the neurons that are affected in Huntington's disease. So it's a selective degeneration. And other work has been done that I don't have time to summarize, but suffice it to say that the evidence to support a problem with the mitochondria in Huntington's disease is rather strong. Now, as I also mentioned, you see nuclear inclusions in these disorders, and it turns out that if the mutant protein can't get into the nucleus, you cannot have pathology. And so that led people to look at transcription, which is the regulation of gene expression, and a number of studies have shown that altered transcription, transcriptional dysregulation, appears to be a key event in Huntington's disease. And so these two different pathways have been implicated, but the problem has been, to reconcile them, because the mitochondria is quite distinct from the nucleus. Now, our work on Huntington's disease has involved a certain mouse model known as the N17182Q mouse model. This is a model that was generated by David Borchardt when he was at Johns Hopkins University. And these mice produce just a fragment of the Huntington's disease gene, but it contains the mutant polyglutamine stretch, and they show a phenotype that involves weight loss. They have motor symptoms beginning usually around 16 weeks of age, and the mice succumb to the disease anywhere from 20 to 25 weeks of age. And so we've been using these mice for our studies, and we decided that we should look at how these mice were regulating their body temperature. And admittedly, the rationale for that was we wanted to get baseline information as a prelude to a preclinical trial that we received funding for. And the way that we measured body temperature in the mice is that we implant a chip between their scapulae, and this allows you to remotely record the body temperature so you don't have to handle the mice, because if you handle the mice, that upsets them, that could alter the measurement. And so that's shown here. And what we found, and it was a rather shocking result, is that mice with Huntington's disease have severe hypothermia. Now, the regulation of body temperature in mammals is very tight, so you should have very little deviation from normal body temperature, as I think everyone knows. And what we found is that when the disease began, there was already the inkling of a significant difference in body temperature, and as disease progresses, it becomes quite pronounced, this difference, and to the point that it's quite abnormal, so that many of the mice have body temperatures below 34 degrees Celsius. And here you see traces for individual mice, and what you can appreciate that some mice, their body temperatures drop below 30 degrees, and they're still alive with body temperatures that low for sometimes a week or two. But the mice are still alive with these body temperatures that are quite abnormal. So we first wondered, if this was just something that would occur in any mouse that was destined to die from a neurological disease where there would be extensive muscle wasting. And so we took the ALS mouse model and we measured mice, premorbid mice, for their body temperatures, and we found there was a minor decrease, but it was not significant, and certainly nothing like you saw in the Huntington's disease mice. So that led us to conclude that the hypothermia is specific for Huntington's disease. And then the next thing we did is we wanted to study this a little bit more in a more directed fashion. So we did what are known as cold challenges. And in these studies, what you do is you place the mice at 4 degrees in the cold room, and then you see if they can regulate their body temperature. And what we found is that while control mice can regulate their body temperature, even mice that overexpress the Huntington protein but with a normal glutamine track that these mice can regulate their body temperature normally, but the Huntington's disease mice cannot. And even as early as 12 weeks, you see a problem with body temperature regulation. Now, once disease is more progressed, that becomes much more pronounced, so that very little time, in the cold room, and then the mice show an abnormal body temperature. So this was an interesting finding, and we decided we would present it at a national meeting. And we were pleased to see that it was well received by Nancy Wexler, who's been involved in the Huntington's disease field. She was just here at our institution and gave a lecture at the Genome Sciences Symposium. Maybe some of you saw it. And this is Patrick Waite, the postdoc, who has been involved in this work. He actually now is in Germany and has a faculty position there. And in addition to Patrick Waite, this work was a partnership with another postdoc in the lab, a very talented postdoc, Victor Pineda. So the work I'm going to present is their work. Okay, so the observation is abnormal body temperature. And so where do we go with that observation in terms of understanding its molecular basis? Well, as it turns out, body temperature regulation in rodents has a unique feature in that there's a specialized organ for generation of heat, and that's called the brown fat. And so there are various locations throughout the body where rodents have brown fat. Humans have some brown fat when we're infants, but then we lose our brown fat. We're left with just a little bit in our axilla. So when we get cold, we can only rely on shivering to generate heat. We do not have brown fat to keep us warm. And the way temperature regulation occurs is that cold is sensed in the brain, in the hypothalamus. You get a sympathetic outflow. And so if you're a rodent, then what happens is the sympathetic outflow is received by beta-3 adrenergic receptors that are on the brown fat cells. And then what should happen is you should have upregulation of a transcriptional coactivator known as PPAR gamma coactivator 1 alpha, or PGC1 alpha. And then PGC1 alpha is involved in driving the upregulation of uncoupling protein 1, or UCP1, and also in driving the production of mitochondria. And uncoupling protein 1, what it does is it uncouples the oxidative phosphorylation pathway in the mitochondria. So instead of generating energy, ATP, you have a Feudal cycle and you generate heat. And PGC1 alpha is felt to be important for the brain as well as the brown fat. And so we thought, okay, this is something that we should study in the Huntington's disease mice. And the first thing we did is we looked at the brown fat from the Huntington's disease mice, and what we found is that Huntington's disease mice have brown fat that doesn't look like brown fat at all. It has reduced cellularity and has many lipid vacuoles, so it looks really like white fat. So the first thing is that the brown fat of Huntington's disease mice is abnormal. And we wanted to, of course, check if the Huntington transgene is expressed in brown fat. So this experiment shows when you do a PCR experiment and you try and amplify the messenger RNA that it is indeed there in the transgenic mice. So you know that the protein is being expressed in the brown fat. Okay. And so what we did is we compared mice that were at room temperature. Those are the solid bars. And then the thatched bars are the mice that have been placed at 4 degrees for a number of hours. And the first thing we did is we said, well, are they able to upregulate PGC1 alpha appropriately? And as you can see here, HD mice do just fine. The upregulation of PGC1 alpha is comparable to controls. However, when you look at UCP1 at the RNA level, you see a marked reduction of UCP1 in mice that are cold challenged. And here you see a Western blot for UCP1, and again, you see that it's just not being upregulated. So while PGC1 alpha is appropriately upregulated, the UCP1 response is blunted. So the next thing we did is we said, okay, well, is this a problem with the transcription regulation? And so in this experiment, we took pre-adipocyte cells, and we put in a reporter. So the promoter of the UCP1 gene is linked to luciferase, which allows you to measure the promoter activity, if you will, the transcriptional activation. And in this experiment, what we did is we transfected in normal Huntington protein. It's a fragment of the protein, normal in that the glutamine length is not expanded. And what we found is that when you stimulate with a ligand that would drive expression through using the PGC1 alpha coactivator, that you get a nice activation of this promoter reporter. However, when you put in the mutant Huntington fragment, you get a severe inhibition of this activity. But what we observed is that if you co-transfect in PGC1 alpha, you could rescue that inhibition. So that suggested to us that the transcription interference involved the PGC1 alpha. And so the observations that we made in the mice, obviously were very interesting. But an important question is, well, is this relevant to Huntington's disease in human patients? So the way we addressed that question is we obtained material from human patients, Huntington's disease patients, from the region of their brain that's affected, the striatum, and we focused on individuals that were either pre-symptomatic or very early symptomatic for Huntington's disease. The scale here goes from 0 to 4 in terms of the grading, so these are asymptomatic or early symptomatic. We had striatal material from controls, and in this experiment, what is done is it's called a microarray comparison. So in this experiment, what we're looking at is genes that are regulated by PGC1 alpha. So we identified a set of 26 genes that are upregulated by PGC1 alpha, and then we asked, well, in the brains of Huntington's disease patients with those genes, how would they compare their expression levels to what we would observe in controls. And so here in this histogram, where red is up and green is down, what you can appreciate is that in the Huntington's disease patients, many of these PGC1 alpha target genes are downregulated. And so that told us that perhaps in human Huntington's disease something is awry that involves PGC1 alpha pathway. And so just to elaborate upon this a bit more, for those of you who are microarray aficionados, the approach that we took is called gene set enrichment analysis. And this is a statistical method that allows you to evaluate a functionally related sample set. And as I said, we looked at these 26 target genes, and what we noted was a highly significant coordinate downregulation. So this really indicated to us that the PGC1 alpha pathway is abnormal in human Huntington's disease patients. And so the observations in the mice may very well have relevance to what's going on in the human patients. And then what we did is we simply validated the results. What's shown here is that the genes that we found that we identified as being decreased were indeed decreased. And these are our controls. You should have a decrease in dopamine receptor D2. You should have an increase in GFAP. And so this was just simply the validation of the data, because even though you see it on the microarray, you still need to follow up and validate that result. And then a final experiment that we did, because we wanted to consider whether these PGC1 alpha changes really had functional significance at the level of mitochondrial activity. And so what we did is we obtained brain slices from our mice. And, there's someone's pager. Someone did not follow the rules, did they? So what we obtained is the brain slices from the Huntington's disease mice and from control mice. And using a system for measuring oxygen consumption, a flow-through system, we were able to see if oxygen was consumed appropriately in the face of being exposed to different substrates. And what we found was that when you exposed these brain slices to lactate pyruvate, you had a significant decrease in the ability to consume oxygen. And that's shown sort of here with this little blip difference. And so that was an interesting finding, especially since the LDH enzyme is regulated by PGC1 alpha. It was consistent with the microarray finding. And what it said to us is that, sure enough, there is a mitochondrial abnormality that results from the PGC1 alpha transcriptional abnormality. And so that was our conclusion in a paper that we published about 1.5 years ago. And so, as I discussed at the outset, people were having a hard time reconciling the mitochondrial abnormalities, which most people feel are very important for Huntington's disease pathogenesis, with the nuclear transcription abnormalities. So, people used to think of this, that these were two events that were occurring in parallel and then were converging, but really unrelated events. But then based upon our data and others, people now feel that there is a relationship. And so what's going on is that there's a nuclear transcriptional abnormality that involves PGC1 alpha, and that is very much playing into the mitochondrial abnormalities that are likely a key contributor to Huntington's disease pathogenesis. Okay, and so where are we taking this work in 2008? Well, we're attempting a genetic rescue by crossing the Huntington's disease mice with transgenic mice that inducibly express PGC1 alpha. That's work that's being done by Taiji Tsunemi, a fellow in my lab. And we also are looking for PGC1 alpha dependent transcription factors that might be the exact target of the polyglutamine expanded Huntington protein. And that work is being continued by Victor Pineda. Okay, so now I'm going to transition to my next story, which is SEA7. But before I do that, I do want to tell you that I had never been to Paris until a month ago, and I was fortunate to be able to go there to present my Huntington's disease work at a very nice conference. And I did want to go up the Eiffel Tower, but unfortunately, the day I was there, the Olympic torch was making its way through Paris. And as I approached the Eiffel Tower, I saw lots of people. I said, wow, I didn't realize the Eiffel Tower would be so popular in April. And the reason why was there was some protest activity, as you can see here. So I guess all that means is now I have to go back to Paris next year maybe and see the Eiffel Tower. It was a great trip, even though I didn't get to go up the Eiffel Tower, I can assure you. Okay, so now I'm going to tell you a little bit about some of the work going on with Spinocerebellar ataxia type 7. This is a disorder that we call SEA7 for short. It's an autosomal dominant cerebellar ataxia, and it's recognized clinically because patients also show retinal degeneration. We won't really discuss the retinal degeneration too much today. The gene was originally mapped to chromosome 3P, and it was cloned in 1997. It's a novel gene known as ataxin 7 that encodes an 892 amino acid protein of previously unknown function, but we've helped to figure out what its function might be. We think it's a transcription factor. The CAG repeat starts early on in the protein coding region, and it's a polyglutamine disease, as we've discussed. I just want to point out that what's interesting about this disorder is you can get very large repeat expansions, and sadly, it's not uncommon to have childhood or even infantile onset cases of this disorder. Now, our goal in terms of approaching understanding this disease was to model it in mice, and we thought this would make really good sense to try and generate an animal model. And the question was, well, what model system should we use? And we decided we would stay away from the crossbred flying fish piranha model that Professor Zerkowitz made the unfortunate decision to pursue. Probably it would have been cheaper, though, than keeping mice. But we went with mice. And so our plan was to develop a mouse model for SEA7. And our approach for this particular project was to take a well-worn pathway, and that is to use this murine prion protein expression system developed by David Borschel, and to introduce two different cDNAs, both of them ataxin 7 cDNAs, but one with a normal repeat, CAG24, and one with expanded repeat, CAG92, and to derive lines of transgenic mice. And so we took this approach, and we ended up with 4 different 92Q founders and one 24Q founder. And when you do this type of experiment, you want to quantify the expression level, and that's what we did. The approach that we use is an RT-PCR co-amplification strategy, so where we measure the transgenic human ataxin 7 expression and compare it to the mouse, and that's what these ratios represent. And what I want to emphasize is we were fortunate in that we obtained 2 lines, an expanded line and a control line that had very comparable expression levels. And so that's important because it allows you to compare those lines and draw conclusions in terms of what can be attributed to the repeat expansion. And so that's how we proceeded with this line of investigation. And once you know you have proper expression levels, you want to look at the spatial pattern of expression. And so here you're looking at brain sections from transgenic mice, and we're labeling the protein with an antibody directed against ataxin 7. It's an antibody that we developed in our lab. And you see regions of the brain that are important for spinocerebellar ataxia. You see the brainstem here in panel E, and this is the basis pontus. These disorders are also called olivopontocerebellar atrophies. So the pontus is important, and this is actually the inferior olive, the olivo. So these are regions where you see expression of ataxin 7 protein, and that's what you would hope for. And if you go in at high power, you can see these inclusions that are in the nucleus, these very intense staining aggregates that are in the nuclei of the neurons. And here we see a 24Q control for comparison, where you do not see accumulation of misfolded protein. So this was very encouraging. And before I proceed, I do want to tell you also that we studied the mice in terms of their phenotype, and they developed an SEA7-like visible phenotype. The first sign of that was a gait ataxia, so they would have a funny walk, and that would begin anywhere from 8 to 17 weeks of age. This would progress until the mice really became severely affected and would even fall over when you observe them occasionally. And ultimately the mice become tremulous, they got a whole body tremor, and they died prematurely. And we really didn't have an obvious cause for death at that time. And so this was what came out of the initial work. And I do want to point out that Bryce Sofer, a wonderful research scientist who's been in my lab since pretty much its beginning, has been involved in this project. And a lot of the work that I'll discuss today was done by Sarah Custer, who was a Ph.D. student in my lab, graduated, and now is a postdoctoral fellow at the University of Pennsylvania, a fine institution. Dan, wouldn't you agree? We studied these mice, and we made a really unexpected observation, a very interesting observation, and that is that you would see a degeneration of Purkinje cell neurons without expression of ataxin 7 in those Purkinje cells. So let me take you through this slide. It's very important. It's a beautiful image of the cerebellum, and let me just review the neuroanatomy of the cerebellum with you. So the way that the cerebellum is organized is you have granular cell neurons that are out here in the granular cell layer. You have Purkinje cell neurons whose cell bodies and nuclei are here in the Purkinje cell layer, and their dendrites are found in the molecular layer of the cerebellum. There's also other neurons in the molecular layer and non-neuronal cells, as we'll discuss, glia. And so here we're staining the Purkinje cells with an antibody against calbindin, because calbindin is highly expressed in Purkinje cells. That's what gives you the green color. And nuclear compartments are stained with DAPI, and that's what gives you the blue. And then we're also using an antibody against ataxin 7 that labels with the magenta color. Of course, there's no ataxin 7 overexpression in, or human protein expression in the non-transgenic mice. But what you see in the transgenic mice, what I want to point out is you have this pronounced Purkinje cell degeneration, as I think you can appreciate. The dendrites are totally atrophied, and you have decreased staining of calbindin, and it just really is a very abnormal appearance. And what you see is that ataxin 7 aggregates are found in pretty much all neuronal nuclei in the granular cell layer and the molecular layer, but there is no expression of ataxin 7 in Purkinje cells. And so that was a peculiarity of the expression system that we used. And what this shows is that even without expression of ataxin 7 in Purkinje cells, the Purkinje cells degenerate. And so this process of degeneration is called non-cell autonomous, in that it involves a different cell. And so this was a very interesting observation that we wanted to pursue, what the basis of that would be. And one of our candidates for involvement in this process are the Bergman glia. Well, what are the Bergman glia? Well, here again we see the cerebellum, and the Bergman glia here are labeled in green because green labels glial fibrillary acidic protein. Purkinje cells are in red with the calbindin. And Bergman glia are a resident population of glia in the Purkinje cell layer of the cerebellum. You know, our nervous system doesn't consist of just neurons, there's many other cell types. And so glia are cells that their importance really has been underappreciated for decades, but within the last decade or so, people have come to appreciate that they have important functional roles. And in the cerebellum, Bergman glia are especially important. Their cell bodies surround Purkinje cells, their radial processes ensheath the Purkinje cell dendrites, and they provide important trophic support, and they remove glutamate from the synaptic cleft. Glutamate can, when not removed, cause toxicity, as we'll discuss. So, to pursue the basis of the Purkinje cell degeneration, we did ultrastructural analysis using electron microscopy. And what I want to show you here is that our SCA7 transgenic mice had abnormalities, and at high power what we saw was there was increased electron density and swollen endoplasmic reticulum. That's abnormal. But one thing that we also appreciated was that the Bergman glial fibers were no longer tightly opposed to Purkinje cell dendrites, so you would have these gaps. And those gaps are abnormal. And so that was the first suggestion, that the Bergman glia were abnormal. Now, you know, again, this is in the mice. I always emphasize the importance of trying to make this relevant to the human disease process. We were fortunate in that we did obtain human SCA7 brain material, and when you stain for GFAP, what you see is that there is some Bergman glial pathology. This is an abnormal pattern of reactivity compared to control brain. So it did make us feel we were barking up the right tree, so to speak. So we decided that, okay, we have evidence suggesting Bergman glia is important, so let's test this in a directive fashion. So the question we wanted to answer is as follows: is the expression of polyglutamine expanded ataxin 7 in Bergman glia sufficient to cause neurodegeneration in mice? And so our plan was to generate a transgenic construct. We used a promoter known as the GFA2 promoter. This is a 1.2 kb fragment of the human GFAP promoter, and it drives robust expression in Bergman glia in the cerebellum. And our plan was similar to the plan that I explained for the PRP transgenics, and that is to use two different allele sizes. In this case, we used 10q and 92q, and to try and obtain lines where the expression would be comparable to one another. And so that's what we did. We generated those lines of transgenic mice, and then we set out, to study them. And the first thing we did is we wanted to see, again, the spatial expression pattern. And so here you see sections from the cerebellum, and in the top panels, the green corresponds to the ataxin 7 labeling, the red to the calbindin, and you have non-transgenic here, 10q and the 92q. And what you can see is that in the transgenic mice there's expression of ataxin 7, and the ataxin 7 is accumulating around Purkinje cells. Red is calbindin to indicate the Purkinje cells. You can also already appreciate that these cerebella are not normal. And then to be a bit more precise about this, we tried to co-localize the expression of ataxin 7 with GFAP, and what you can see is sure enough that you have expression in Bergman glia cell bodies, which is what we were aiming for. Now, were the mice normal? Well, they were not. They did develop a progressive ataxic phenotype. And here you see some of the behavioral testing results. Clasping is an abnormal response where when you suspend a mouse by its tail, it withdraws its limbs in instead of splaying them out, which is the normal response. And the ledge test is just a balance test that we do to see if the mice can stay on the side of the cage without a problem. And they failed those tests pretty miserably. The 92q individuals did. And then the rotorod is a rotating rod, almost like spinning on a log, if you will. And so the mice are put on that, and we see how long they can stay on there. And what you can see is that individuals that were expressing the polyglutamine expanded ataxin 7 in their Bergman glia performed much worse than individuals who either expressed the normal ataxin 7 or were non-transgenic. So the mice developed an ataxic phenotype similar to what you would expect in patients with SCA7. And did want to point out that unlike the PRP model, the ataxia is an isolated phenotype. The mice live a normal lifespan. So you have pathology that's restricted to the cerebellum, which again makes sense when you think about how this experiment was designed. Now when you look at the cerebella of the 92Q mice and you use the Calbindin staining approach, you again see abnormality. You see reduced Calbindin immunoreactivity, and you also see that the molecular layer is a bit shrunken. So this is abnormal. So there's abnormal histopathology. And if you look at the Bergman glia themselves, again using GFAP, you see that the staining is abnormal, so that you have swollen Bergman glial fibers and very reactive Bergman glia in the 92Q mice that are shown here in panel C in comparison to control transgenics and non-transgenics. And also, this was interesting because if you look at human patients with SCA7, you see a similar pattern of swollen Bergman glial fibers. Now, of course, an important question is, well, what's the molecular basis of the abnormality? And so there, we decided we would look at the expression of a Bergman glial specific glutamate transporter GLAST. Now, as I mentioned, one of the important roles of glia is to remove neurotransmitters from the synaptic cleft. Because if you have neurotransmitters hanging around too long, the neuron gets excited over and over again, and that repeated excitation can lead to problems with calcium flux and ultimately to the death of neurons. And there are a number of different glutamate transporters that are important for removing glutamate from the synaptic cleft. And as it turns out, Bergman glia in particular express GLAST. And we decided that we would look to see what the expression level was of GLAST in the 92Q mice. And we also looked at GLT1, an important glutamate transporter that's expressed in neurons, as well as glia, and EAT4, which is expressed by Purkinje cells. And what you see here is there was a reduction in the level of GLAST RNA expression in the 92Q mice, but the GLT1 and EAT4 expression levels were unaltered. And this was confirmed by Western blot analysis at the protein level, where you see a reduction in the protein level of expression of GLAST in the cerebellum. And I want to point out that Bergman glia are really the only source of the GLAST glutamate transporter in the cerebellum. So this reduction would be linked to the Bergman glia cell population specifically. And I think some of these points I've already made. I should point out that in these experiments, we are looking at pre-symptomatic mice, because the concern is, once the mice have a phenotype, then you may have abnormalities that are non-specific. So we always look at mice before they have a visible or even a rotorod abnormality. Okay, and then the next thing we did in this study is we decided we would take Bergman glia from the mice and culture them and directly study the glutamate transporters. And what you see here is that when you culture Bergman glia from the mice and you can measure the glutamate transporter expression and function, and GLAST expression is reduced, as shown here at the RNA level and at the protein level, GLT1 expression is unaffected. And we also performed glutamate uptake assays, and that result actually is shown here. And we performed that, I don't want to get into the neuroscience here too much, but what we needed to do is we needed to isolate the GLAST contribution. So we used a pharmacological agent, dihydrocanate, that blocks GLT1, so we could isolate the GLAST contribution. And so in this glutamate uptake assay performed in Bergman glia, we saw reduced glutamate uptake function in SCA7 Bergman glia. And then the final series of experiments that we did related to studying glial glutamate uptake are shown in this slide. And what we did first is we did cerebellar slice assays, where we generated cerebellar slices again from the 3 different types of mice, and we saw a trend toward a reduction in glutamate uptake. It wasn't significant, but it was a trend. And then we did what are called cerebellar synaptosome preps, where you isolate the cerebellar synaptosome that contains the synapse region, and look for uptake of glutamate. And what we found is that in the 92Q individuals, there was a significant reduction in glutamate uptake capacity. And the final experiment that we did along these lines was a functional experiment. And in this experiment, we took granular cell neurons from wild-type mice, and then we co-cultured them with Bergman glia that we obtained either from transgenic mice or from control mice. And then what we did is we said, okay, let's expose these neurons to glutamate and see if the Bergman glia will protect from this glutamate stress, because as I said, glutamate is a neurotransmitter that will lead to neuron cell death. And as you can see here, when there's no co-culturing, the amount of death is significant. But when you co-culture with non-transgenic or controlled transgenic Bergman glia, you have protection from the glutamatergic stress. However, co-culturing with the 92Q Bergman glia yielded susceptibility to the glutamatergic stress. So that would be a functional correlate saying that, well, the problem with glutamate transport is a significant event in terms of neuropathology, potentially. And then another set of experiments that we did to sort of nail this down for ourselves is to ask the question if the Purkinje cells were dying in a way that you would expect if there was glutamatergic stress. And the way the Purkinje cells die when they're under this type of stress is through what's called dark cell degeneration. So we simply used the electron microscopy to look for dark cell degeneration in the SCA7 transgenic mice, and we found morphological features that were consistent with this pattern of degeneration. And just tell you a little bit about the future directions that we're taking. We're very interested in therapy development, so we're trying to identify compounds that can boost GLAST expression levels as an idea that we could test them in mice, with the idea that ultimately we might be able to use them in patients who have this disease. And we're also generating an SCA7, we've generated it already, an SCA7 mouse model that has pan-neuronal and pan-glial expression with the flock cDNA, so that we can remove that cDNA from different cell types in the central nervous system to test the question of necessity, which is the flip side of sufficiency. Because what we've shown is that, sure, if you express mutant ataxin 7 in certain cell types, that's bad. But now let's go back and let's remove it from certain cell types and see, if that will make a difference.